speaker night with Professor Jennifer Doudna, presented by MCB USA. My name is Daryl. I'm one of the co-presidents of MCB USA. So briefly about MCB USA, we're the original MCB department-sponsored club at UC Berkeley, and we aim to facilitate the formation of relationships between students and faculty, and students and other students, along with exploration of biological careers in health and industry. And we do this by providing a lot of valuable resources and hosting a variety of events, such as Speaker Night tonight. And we strive to promote the professional and educational development of all biology students at Berkeley. So now I'd like to turn the mic over to our event committee co-chairs, Carmen and Anissa, to talk briefly about our upcoming event, which is Emphasis Night.
Hello, I'm Carmen, and we just want to let you guys know that this Wednesday, March 16th, from 7 to 9 in 122 Wheeler, we're going to be hosting Emphasis Night. This event is for any of you who are thinking about majoring in MCB or are unsure of which emphasis you want to go into. Come to our event to hear faculty advisors and professors such as Robert Beatty from Immunology and Dr. David LaRue from Neurobio come speak about major upper division classes in each emphasis, future career paths that are possible from each emphasis, recurrent research going on in each field, and answer any questions that you guys might have. Just a reminder, it's March 16th, 7 to 9, and we will have like some time at the end for one-on-one time. We just wanted to get it, so bye.
Now Simon, our Education Committee co-chair, will introduce Professor Jennifer Doudna.
Hi everyone, my name is Simon Arfei. I am the Education co-chair of MCB USA, and I would like to welcome you all to our Spring 2016 Distinguished Keynote Lecture Series. Being part of the Berkeley community is always a great honor to learn about cutting-edge research, and our speaker tonight is an exemplar of what it means to do cutting-edge research. So on that note, let's introduce our speaker tonight. Professor Doudna currently holds a joint professorship in chemistry and molecular cell biology at the University of California, Berkeley, and has been an investigator at Howard Hughes Medical Institute since 1997. Professor Doudna's research has been focused on the elucidations of the functions of catalytic and non-protein coding RNAs. Specifically, she has actively engaged in deciphering the molecular structures of biochemical activities of RNA enzymes under Tom Cech, where she did her postdoctoral fellowship, and Tom Cech was a recipient of the Nobel Prize in Chemistry in 1989. The Doudna Lab is currently focused on three main projects. Number one, internal ribosome entry site RNAs. Number two, RNA recognition by dicer enzymes. And number three, CRISPR systems and bacterial immunity, which will be related to the topic today. Professor Doudna, as we all know, has been a recipient of numerous accolades and magnificent accomplishments, including the Breakthrough Prize in Life Sciences, the Alan T. Waterman Award, the Lurie Prize in Basic Biomedical Sciences. Professor Doudna is also a member of the National Academy of Sciences, American Academy of Arts, Sciences, and National Academy of Medicine, and has been called a citation laureate by Thomson Reuters magazine. So we will be expecting Professor Doudna to be winning a Nobel Prize in the near future, based on the predictions of the world. So without further ado, it is a great honor and privilege to introduce our speaker tonight, Professor Jennifer Doudna.
no scientists in my family, grew up in a small rural town in Hawaii, and just by the process of following my my interests and my passions, and I had a few key mentors along the way, I sort of find myself standing here today. So I don't want you to think that you kindly gave me this very generous introduction, but I really don't want you to think that we're any different. I think that all of us have a passion for science, and we have a sort of shared community here, and I really want to communicate that tonight. That for me, science has always just been about the process of discovery. The story that I want to tell you tonight really has extremely humble beginnings. It was the project that started in my lab here at Berkeley as probably, I thought, one of the most obscure things we'd ever worked on. And I certainly never thought it would turn into the technology that it has become. So I'm going to share with you that process of discovery. I want to tell, because we're all scientists here, I want to share with you a little bit of the real-time science that's going on in the lab right now, what we're trying to do. I have a very active group of researchers that include actually a large number of undergraduates, and we always have, so that's been great. And I'll try to tell you how this technology that is that I'll describe tonight about from our fundamental research project. So the story of CRISPR in my lab really began about 10 years ago with a phone call that I got from Jillian Banfield, who's a colleague of ours here in College of Natural Resources. Her lab investigates bacteria growing in environments in nature where they often are not, we haven't been able to culture them, so we don't really know what bugs are there. And her lab collects samples and sequences all the DNA in the sample, and then reassembles those sequences to figure out the genomes of the bugs that are growing in those environments. And she called me one day in my office here at Berkeley, and she said, we see something very kind of strange in our DNA sequencing data, and I think I need to tell you about it. So we met for coffee at the Free Speech Movement Cafe, and she shared with me the fact that in a lot of their sequencing data, the bacterial chromosomes had a very unusual repetitive DNA sequence. And it had a pattern like this. Let's see, is this a pointer? Come on. Find anyone. I don't know if I have a pointer, but if I don't, maybe can I use my mouse? Yeah. Okay. So basically it had a series of repetitive DNA sequences, and these were about 40 base pairs long, repeated over and over, the genome, and in between the repeats were unique sequences of about the same length, shown in these colored boxes, and in 3 publications in 2005, labs had recognized that these spacer sequences in between the repeats were sequences that matched exactly DNA sequences from viruses that infect these bacteria. And so this was noted, and there was no experimental data at the time, but this made a few labs start to think, I wonder if this is some kind of adaptive immune system in bacteria, some way that bacteria can make a genetic record of the infectious agents they've encountered over their history, and kept a little piece of those viral sequences stored in the genome for maybe future protection from those infectious agents. And a couple more things about this. So these had been called CRISPRs because of the properties of these repeated sequences. And next door to the CRISPRs were very typically CRISPR-associated or Cas genes. So it made it look like this might be some kind of system in bacteria, a conserved set of genes and sequences that were being used for some purpose. So why did Jill get in touch with me? Well, she knew that my lab was researching RNA molecules, and we have a long-standing interest in how RNA molecules control the way that genes are expressed in cells. And she thought we might be interested in investigating the properties of this proposed pathway. So I'm going to share with you 3 things tonight. I want to tell you, first of all, a little bit about how our studies, and with many others, of course, studies of these systems became a technology for genome editing. And then I'm going to say a little bit about the research that's going on in our lab right now to figure out how these genome editing molecules really operate in cells. And then finally, I want to tell you a little bit about a different aspect of CRISPR pathways that enable cells to acquire new sequences into the genome, how that really works. So the experiments that kind of got started probably in the mid-2000s, around 2005, by just a handful of labs around the world were focused on testing this hypothesis. Are these repetitive sequences really an adaptive immune system in bacteria? And so what was done was to make mutations in the Cas genes, and then ask whether bacteria that had mutated Cas genes were susceptible to viral infections or not. And in work that was done by two scientists at a yogurt lab, a company that makes yogurt and other food products, they were doing these initial genetic experiments, Philippe Horvath and Rodolphe Marengo. And they were the first scientists to publish their data showing that these CRISPR systems are in fact protective against viruses. So they are immune systems in bacteria. And what emerged in experiments then from multiple labs was that bacteria that have a CRISPR locus have the ability to recognize foreign DNA that gets into the cell, acquire small bits of that DNA into the chromosome in these CRISPR loci, and then generate an RNA copy of the CRISPR sequences that includes these bits of DNA derived from viruses in green boxes. And then those RNA molecules are chopped up into smaller pieces that each have one bit of sequence from a virus in the middle. And those RNAs assemble with proteins encoded by the Cas genes to form protein RNA targeting complexes. And these complexes search through the cell. They're sort of surveillance complexes, and they look for DNAs that match the sequence of the viral sequence in the RNA in the complex. And when they find a match, the RNA and DNA base pair together and allow the proteins in the complex to cut the viral DNA. So it's a great way that bacteria can steal bits of DNA from viruses and then use that information against those viruses to protect the cells. And so in our lab, we started after that free speech movement cafe coffee hour with Jill Danfield. We started studying this pathway, and in my lab we do a lot of biochemistry. So what we do is we typically will purify molecules in the lab and then study their function in isolation outside of the cell. And we also do some cell-based experiments too, testing how cells operate when they have mutations in various proteins or RNA molecules. And so we started studying these pathways, and we investigated initially the sort of central part of the pathway right here that allows cells to make these RNAs and form targeting complexes that can bind and cut DNA. So we started studying how that works. And that led to a collaboration with a colleague, Emmanuelle Charpentier, who is a medical microbiologist. So she has a very different expertise from my own. She was studying the flesh-eating bacteria, if you've heard that, called Streptococcus pyogenes, which turns out to have a unique type of CRISPR system in the genome that has a single gene called Cas9 that had been, in 2011, when we met at that time, had been shown genetically to be a critical gene for protecting the flesh-eating bacteria from viruses that might otherwise kill them. And so she was studying this, and we met at a conference, and she said, I think it would be really interesting to know what the function of that encoded protein of this Cas9 gene really is. How does it operate? And so we decided to collaborate to figure out the answer to that question. What's the function of Cas9? What does it do? And so I came back to my lab here at Berkeley, and I enlisted the work of Martin Jinek, who was a postdoc in my lab at the time, and an undergraduate, Nikki Hauer, who was here for the summer, doing the summer project. And he was kind of looking for something interesting to do. And so I said to these two guys, would you guys like to work on this collaboration to figure out what this Cas9 protein is doing? And so they were game. And so they teamed up with Chris Chylinski in the Charpentier lab, And over the next several months, they figured out that Cas9 is a really amazing protein that in this cartoon is shown in the blue sort of blobs. And we figured out that it's a protein that has a unique ability to bind double-stranded DNA, which is this black sort of bubble molecule. And when it binds to DNA, it interacts at a region that has base pairing complementarity to a 20 nucleotide sequence in an RNA molecule. And that RNA is this strand right here that comes from the CRISPR locus. And so in bacteria, this 20 nucleotide sequence would be a sequence from a virus. And so when this interaction occurs, the DNA unwinds in the targeted region and allows the protein to cut both strands of the DNA. So it makes a very precise break in the DNA double helix at this position. And as we were studying this, what Chris and Martin Janek and Mickey Howard figured out was that in addition to the protein and this CRISPR RNA, to cut DNA, this system needed a second type of RNA called tracer, which is the red molecule shown here. And that RNA base pairs with part of the CRISPR sequence to form an RNA-RNA structure right here that is bound by the Cas9 protein. So you have to have those two RNAs to assemble with the protein. And once that occurs, then this protein with these two RNAs forms a very effective DNA targeting and cutting little machine. And so once we had that information, Martin was in my lab and in my office, and we were going over his data. And we looked at the data that he had, and we realized that we could probably simplify the system compared to what nature has done. And the idea that we had was to link together the CRISPR RNA and this second RNA called tracer to form a single guide type of RNA that was sort of engineered in the lab to have the binding sequence for Cas9 recognition right here and the DNA targeting sequence here. And we thought this would create a simple two-component system, one RNA and one protein, that could be easily programmed to bind and cut essentially any DNA sequence by just changing the sequence on the 5' end of the RNA so we can target different sites in DNA molecules. And Martin did a super simple, and it seemed to us, very simple experiment that tested this idea, but one that really kind of gave me chills when I saw the result because of how we suddenly realized that this very kind of interesting but obscure project in our lab might actually be harnessed as a really exciting technology. So here's the experiment that he did. He basically took a double-stranded DNA molecule called plasmid, and looking in the plasmid sequence, he designed 5 different versions of the single guide RNA that would target the sequences that are marked by these red bars. And note that in each case, the only criterion for selecting the sequence that was targeted was to choose a site that was adjacent to a GG dinucleotide in the DNA. So that was a little signature motif that we had discovered during our biochemical experiments to be critical for DNA target recognition by this system. So we picked these sites, designed the single guide RNAs that would provide base pairing capability to those sequences, and then to do this experiment, we took the plasmid and then incubated the Cas9, purified Cas9 protein with one of these single guide RNAs, and then a second enzyme, a restriction enzyme called SAL1 that cuts the DNA about 60 base pairs away from this part of the plasmid DNA molecule. And so these are doubly digested plasmids, and we can analyze the results by isolating the fragments of this digestion reaction on an agarose gel system. And that's what you're seeing down below. So here's the bulk of the plasmid DNA is running at the top of the gel. It doesn't move very far into the gel system, but these little pieces that are chopped out of the plasmid are smaller, and they run down here. We were super excited to see that they had very distinct sizes that corresponded exactly in length to the size that would be dictated by cutting the DNA at the sequence that was marked by the single guide RNA. So this is when we really knew we had a programmable protein. And I went home that night and I was cooking dinner for my son, and I was thinking about the data, I was thinking over the day, thinking about this conversation, and I just burst out laughing in my kitchen, And my son came up to me and he said, "What's so funny, Mom?" And I said, "We have this crazy protein." something like this, but somehow it had. And just to show you in a little more detail now how we understand that this actually works, this is showing you this is now a 3D printed model, so a model of Cas9 with its guide RNA bound to a blue DNA molecule that was printed on a 3D printer using X-ray crystallographic coordinates for a crystal structure of this that isn't exactly this, but it's kind of this is a model that's based on a structure. It's actually a structure solved by Martin Jinek in his lab now at the University of Zurich. And so what you're seeing here in this picture is the Cas9 protein in white and the guide RNA in orange that sits in the protein such that when this complex searches through the cell and finds a matching DNA sequence, it opens up the DNA helix, this blue double helix here, to allow hybridization of the guide portion of the RNA. So you can see an orange-blue hybridization, a little RNA DNA helix that goes inside the protein and unwinds the other strand of the DNA, and then two active sites in the protein are positioned correctly to cut the two strands of the DNA. So it's like a scalpel, just whoosh, cuts the two strands of the DNA without a double-stranded break. And so you might be thinking, well, that's great if you're a bacterium and you want to destroy a virus, but why is that useful for genome editing? Here's where it's really helpful to have, have the scientific literature at our fingertips all the time, right? The papers coming across my desk and, reading about what other scientists are doing in totally unrelated fields, because I have been seeing a lot of papers from labs that were doing something entirely distinct, it seemed like, from what we were doing, but they were taking advantage of the way that plant and animal cells, eukaryotic cells, interact with their DNA to do genome engineering. Just to tell you very briefly about this, so there was a long history, several decades really, of people investigating how DNA mutations and breaks are repaired in cells. And you probably all know this is an important field for cancer biology and other things like that. And so what had been figured out was that in plants and animal cells, when a double-stranded break occurs in the genome, like this sort of in this cartoon right here, cells have ways of repairing those breaks very precisely. So that can be done by non-homologous end joining, in which the two ends are chemically ligated back together to seal the break, sometimes with a little mutation that is produced at the site of repair, or by homology-directed recombination. You can actually insert a new piece of DNA at the site of the break by recombining in a molecule that would now introduce new genetic information at the site of the break. So the challenge was, how do you introduce a break in a genome at the place where you want to change the sequence? And so there have been a number of strategies for doing this over the years, but the most exciting ones that had come about in the last few years was the ability to make engineered proteins like talons and zinc finger nucleases, and there are a few other examples like homing endonucleases that can bind to DNA very precisely. And by taking those proteins and linking them to DNA cleaving domains, you could have a site-specific DNA cutter. And so it had been demonstrated that you could use these strategies to cut a genomic DNA and trigger very specific repair like this. And we thought, well, I should say that these technologies, although very powerful, had not been widely adopted because it required new protein engineering for every experiment. And we thought, wouldn't it be great if we could take this little bacterial enzyme that can be easily reprogrammed by just adding a different piece of RNA to the protein and use that as the way to introduce targeted breaks to the genome that we then do here. And so we published that idea, and what happened next was really amazing because a lot of labs read that paper and said, that's easy to try. I think I'll test it in my system. And so what emerged over the next few months was that labs around the world started to test this idea in different systems and publish their work. And so we found that this Cas9 single guide RNA system, what it turned out to be a great way to make precise changes to the genome in plants, fungi, mice, fish, rats, human cells, stem cells, I could go on, right? So really a large, it seemed to be very capable of operating in any type of cell. So very useful for genome editing. It also turned out to be could be harnessed for controlling transcription in cells by making inactive, catalytically inactive versions of the protein that don't cut the DNA, but bind very specifically in an RNA-guided way. So you could use them the way you might use something to block the ability of a polymerase to get onto DNA, or recruit polymerases to use genes so you can up or down regulate transcription, and also for imaging in living cells. And I'm going to show you in a couple of minutes an example of doing that. So let me show you a video that was created by Janet Iwasa at the University of Utah for us that illustrates how we might imagine this operates in a eukaryotic cell. So this shows we're zooming into a cell, and of course the eukaryotic cell has a nucleus. And so we have to imagine that this bacterial enzyme has to get into the nucleus of the cell, and then it has to deal with chromatin. DNA is highly packaged inside the cell. And so somehow this RNA-guided protein searches through all the DNA of the cell to find, in principle, a single site in the DNA that has a 20 nucleotide sequence that matches the sequence of the guide RNA. When that happens, it binds to the DNA, unwinds it, like I showed you in that 3D printed model, cuts the DNA, and then hands off the broken ends of the DNA to the repair machinery in the cell, where the DNA can then be replaced or repaired by, in this example, integrating a new piece, a new segment of DNA at the site of the break. And so by doing this, it's been possible to make, to do genome editing in really a wide variety of different kinds of cells. So let me say now a little bit about, so when you see a video like that, obviously it's an artist's rendition of how this might work, but it raises some interesting questions that we can think about scientifically. We can propose hypotheses that we can test in the lab. And so one of the things we started to wonder about is, how does Cas9 find those sites in the genome? And how does it unwind the DNA and make a very precise cut? And that's what we've been working on studying in the lab here since 2012 when we published this work originally. And I'm just going to share with you a few experiments that we've been working on, a few things that we've done to start understanding this process. And let me just tell you a couple of other things about Cas9 that make it a really useful protein for genome editing. And so one is that it turns out to be easily modified. So in addition to being able to make catalytically deactivated forms of this protein that we can use for transcriptional control, as I mentioned, you can also make various kinds of chimeras of Cas9. And that means taking the Cas9 gene and fusing it to some other functional domain, like a domain that recruits or methylates DNA. And so you can then have a site-specific DNA methylase, And so it's been really fun to see how labs have started to adapt this protein for all sorts of applications that are different from what it naturally does in bacteria. Secondly, it's naturally multiplexed. So in bacteria, this is a protein that gets programmed with multiple guide RNAs in the cell to provide protection from multiple viruses at the same time, much like our own adaptive immune systems work. And so as scientists, we can take advantage of that property by programming Cas9 with different guide RNAs for an experiment. So you can make changes to multiple sites in a genome at the same time. And I'm going to show you now that we have evidence that in nature, this is a protein that has evolved for rapid and accurate DNA target recognition. And because of that, that really is what makes it a really useful technology, that it's really pretty good at what it has to do. And so one of the questions that we've been puzzling about is this one here, which is what drives DNA unwinding? So one of the keys to the way this works is that it grabs onto DNA in an RNA-guided way, and then it has to open up the DNA helix to allow that RNA-DNA hybrid to form. And only then does it actually cut the DNA. So, but how does it unwind the DNA? Because if you think of the energetics of that, you've got to pry apart 20 or more base pairs of double-stranded DNA in the middle of, chromatin, right? It's highly packaged. And this is a protein that doesn't use an external energy source. So it doesn't hydrolyze ATP. And so where does the energy come from to unwind the DNA? And we don't really know yet, but I'm going to tell you what we think is going on based on some recent data that we have. And I'm going to summarize this in just a couple of slides. So one of the first things that was done, and this was done by two graduate students in the lab, Sam Sternberg and David Taylor, was to use electron microscopy to visualize the structure of Cas9 as it assembles with RNA and then with DNA. And what those experiments showed us, and I'm just showing it to you here in cartoon form, is that the protein starts off in kind of a closed state, and then it undergoes some kind of a big structural change when it binds to RNA to open up a channel in the center of the protein, which is where the RNA-DNA hybrid ends up. And now, with a series of higher resolution X-ray crystallographic structures of Cas9 in each of these states, we can now put together a movie that models that structural change. And I can show that to you on the next slide. This was a movie created by a student, Ben LaFrance. And so this movie starts off with Cas9 in the closed state, no RNA yet, and you're watching it morphing to the structure that it forms when it binds to the guide RNA, which is in orange. And so here's the central channel in the middle of the protein. And I'm going to show you now what happens when this binds to DNA. So there's an additional conformational change here that allows this protein to accommodate the RNA-DNA hybrid that forms when this assembles with the substrate. And then finally, there's an additional change in the structure right here, which is a domain in the protein that actually cuts the DNA. This is the cutting domain. And we know that it has to swing into place, and it does that only when this is engaged on an actual target sequence. If there are mismatches in the DNA, or that full RNA-DNA hybrid doesn't form, then this final change in the structure doesn't occur, and no DNA cutting occurs. All right, so it's really evolved for this highly coordinated set of conformational changes that allow this to recognize a site accurately and only cut DNA that is bound to the correct sequence. And let's see, so I'm just going to mention that I'm not going to show you the data for this, but all of what I showed you up until now is based on structural studies. And when you do X-ray crystallography, you're trapping the protein in different static states. So, when I showed you this movie, it looks like, wow, we could see those dynamics. We couldn't really see them, right? We're just sort of inferring that they occur by comparing different static structures. But what Sam and undergraduate Matthias Kaplan and several other students in the lab were able to do was to put pairs of chemical dyes on the surface of the protein by using these structures as a guide. They could engineer in pairs of dyes, chemical dyes, that would give a fluorescent signal by chemical interactions between the dyes when they were close enough in space to interact. And so we could use that kind of methodology to detect indirectly these conformational changes. So we actually now have a lot of evidence from those kinds of fluorescence measurements of these conformational states. And we've really been able to confirm that these states are real. And so, just to get back to this question of how does DNA unwinding occur, we currently think that it really comes from this large structural change that happens in the protein. And so you can think of the protein as almost a kind of a spring-loaded protein, right? And when it gets onto DNA, it undergoes this big structural change that basically cranks open the helix. And that we think the energetics of that are somehow very favorable with the protein, and that provides the energy for DNA unwinding. Also, the fact that you're forming an RNA-DNA hybrid, which thermodynamically is more stable in general than a DNA-DNA hybrid, so you get some energetic benefit there. So that's our current thinking. And very recently, two people in the lab, Fubo Zhang and David Taylor, teamed up to use both X-ray crystallography and electron microscopy to determine a structure of the protein bound to an actual DNA, double-stranded DNA molecule, which up until now had not been done. And I'm just going to show you a quick movie of this structure that was published very recently. And so what you can see here is we're zooming into the center of the protein. There's the RNA-DNA hybrid in the structure. And there's the purple strand of DNA that's opened up inside the protein. And by looking at the EM structure, we were able to actually see the ends of the DNA helix protruding from either side of the protein. So we can see that when it binds to the protein, the DNA gets bent compared to what it looks like when it's just in a straight helix. And then this is showing you this rotation of the active site of the protein that cuts this strand of DNA that's bound to the RNA target. And that really puts the protein into an active state, and had not been observed until we were able to trap the structure on a true double-stranded DNA molecule. Previously, that hadn't been seen. So we now know that that final rotation of the active site only occurs when this is engaged on a real substrate DNA molecule. So this slide here just summarizes a lot of data that we had up until last year sometime. That was all in vitro data, meaning from purified proteins that we were working with in the test tube in the lab. And our data really suggested a model in which the Cas9 RNA complex searches through DNA not by grabbing onto one end of the DNA and sliding to find a target, but instead binding and releasing the DNA very, very quickly. And we had evidence that it slows down when it encounters one of these PAM motifs, the GG nucleotide, and that gives it time to test whether there are base pairs that can form between one strand of the DNA and the guide RNA. And if that RNA-DNA can start to form, then the DNA begins to unwind, the RNA-DNA hybrid starts to form, and then eventually, if we get formation of a full RNA-DNA hybrid, then and only then does that active site swing into place and the cleaver cuts the DNA. Right? So, but the question was, what's happening really in cells? Like, wouldn't it be cool if you could actually see this somehow going on in real cells? And so I'm going to show you a couple of experiments that another student, Spencer Knight, has been doing. So he's a joint student between my lab and the lab of Bob Tejan here in MCD. And what we did was to use high-resolution microscopy to start looking at cells that contained fluorescently labeled Cas9, and using microscopy to actually watch the behavior of these labeled proteins as they search through the nucleus of a cell looking for target sites. And I'll just show you a couple of some of the data that we're getting from this. So one of the first things that Spencer saw when he started doing these experiments was that Cas9 has very fast kinetics inside the cell. So here I'm showing you 10 millisecond snapshots of mouse cell nuclei that contain fluorescently labeled Cas9. And so what you're seeing are the fluorescently labeled Cas9 particles moving around inside the nucleus of the cell. And by just comparing the behaviors of these particles, you can see that Cas9 over there on the right is in general moving much faster than what we see for the molecule in the middle, which is a transcription factor, SOX2, and on the left-hand side, a molecule protein that is involved in forming chromatin called H2B, a histone protein. And so that was interesting. We didn't really understand exactly why that was, but it turned out to be very consistent with all of our in vitro data. So this protein has very fast kinetics for reasons that we're still trying to fully understand. And so what Spencer then did was to program Cas9 with different versions of guide RNA that would allow us to compare its behavior when it's searching through the cell using a guide that has a matching site in the DNA versus guide RNAs that don't. And what I'm summarizing here are some of the data for these experiments. And so if you look on the right-hand side, this is sort of shows you how we look at 3D particle tracking in these cells, so we can actually watch a three-dimensional segment of the nucleus as these red Cas9 particles are searching for matching target sites. And this is down here, just showing you the traces of the tracks of those particles. So that's how we actually make these measurements. And then over here on the left are plots that show you the log of the diffusion coefficient on the x-axis and the number of particles with that kinetic behavior on the y-axis. And if we look at that kind of a plot for either Cas9 that's programmed with a nonsense guide, meaning a guide that doesn't recognize any sequence in this genome, or with a guide that has mismatches right next to where the PAM sequence is in the target DNA, these particles generally look, these two plots look very similar. The particles have generally very fast kinetics, and we don't see any evidence that there's stable binding that would slow down the behavior of these particles as they grab onto DNA. But you can see a change when we use a guide RNA down here at the bottom that has a sequence that matches a large number of sites in this genome, 300,000 sites in the genome. This is a repetitive element. And now you can see that a lot of the particles have much slower kinetic behavior. So we know now that that's because they've grabbed onto a matching target sequence and they stay bound to the DNA. And then here's kind of an intermediate case where we have a guide RNA that has some mismatches in the middle of the guide sequence. And now you can see some of the particles that have slower kinetics, but a lot of them are still moving very quickly in the cell. So we don't think most of them are not finding a matching site because they have this mispairing between the DNA and the RNA in that case. And so from this kind of experiment, we can do things like calculate how long does it take Cas9 to find a target site in a big genome like this. And we don't know the answer yet, but we're still making these kinds of measurements with the goal of getting to an answer to that question. And then I want to show you one other experiment using this kind of system, which was to investigate what happens when this bacterial protein that's evolved in cells that don't have chromatin, don't even have nuclei, like bacterial cells, how does it deal with really compacted regions of the genome in a human or mouse cell, namely regions that are called heterochromatin. And so what we did in this next experiment was to use cells that have a fluorescent label on a protein called HP1 that marks heterochromatin, and then watch the tracks of Cas9 particles as they move around these labeled cell nuclei. Doesn't look like much when I show it to you like this, but if we take many, many images and stack them up, what you see is that these very dense sort of fluorescent regions that you can see on this image mark the heterochromatic parts of the genome. And then the colored traces show you the tracks of Cas9. You can see that a lot of the tracks of Cas9 are occurring outside of heterochromatin, but really interestingly, we definitely see forays into these highly compacted heterochromatic regions. And that corresponds to the data that we have from genome editing that shows that we can get editing of sites in the genome that are even within these highly compacted regions. So somehow this protein, perhaps through this ability to grab on and then change its structure when it finds the DNA and unwind it, has the ability to find these sites even when they're buried inside a very compacted part of the genome. So it's a really, really pretty amazing little bacterial machine. So finally, just in the last few minutes, I'm going to tell you a little bit about another aspect of the biology of this system, which is how new viral DNAs are integrated into the CRISPR locus. And so a critical aspect of these immune systems is that they're adaptive. And that means that when a new virus is encountered, the cell is able to find that viral DNA and integrate a little piece of it into the CRISPR locus. And that integration then occurs very accurately. So these new pieces of DNA called spacers are inserted typically on one end of the CRISPR locus, and they go in such that a copy of the repeat is generated on either side of this newly inserted DNA. So you maintain that repetitive structure of the locus. And some experiments that were done by a lab in Israel, in the Weizmann Institute, showed that you just needed 2 proteins from these CRISPR systems to get integration to work in bacteria. And these 2 proteins are called Cas1 and Cas2. So if you had bacterial cells that were overexpressing these 2 proteins only, no other proteins from no other Cas proteins, you could observe integration of new sequences into a CRISPR locus like this. So we thought that this was likely to be the integrase enzyme. And so James Nunez, when he joined my lab as a new graduate student a few years ago, wanted to take on this question of how does this work? And so one of the first things that he did was to start purifying these 2 proteins, Cas1 and Cas2, and he was able to show 2 things that were really cool. One is that they form a complex, so that means that they physically interact, and that physical interaction is critical to this integration mechanism. And he solved the crystal structure of these proteins, and that showed that this is a complex of 6 proteins, 2 types of proteins, but in 6 copies total. And so we have 2 copies of the Cas2 protein at the center in that gold color, and then we have 4 copies total of the Cas1 protein, 2 on each side. So it almost looks like it looked like kind of to us kind of like a butterfly structure. But we didn't know from the structure how this might be grabbing onto DNA and inserting it into the genome. And so to do that, to answer that question, James started doing some biochemical experiments. And what he did was to take DNA molecules that we thought would mimic the substrates for an integrase like this, namely these little double-stranded DNAs that were 33 base pairs long, the length of the spacers that get inserted in the E. coli CRISPR system, where these proteins come from, and incubated them with this integrase complex and a target plasmid that had a CRISPR array in the plasmid DNA. And what I'm showing you down here is an example of the kind of results that James started to get, where we could see that there were products created in these reactions that were covalent changes to the bacterial plasmid DNA that we could see by looking at the changes in, this is now using radio-labeled DNA, so we can actually follow what happens to that DNA during the reaction. So these products occurred that are big, like the size of the plasmid, so we knew that somehow these labeled DNAs were getting chemically ligated to the plasmid. And we found in a series of experiments, I won't run you through all the data, but we found that catalytic activity of Cas1, but not Cas2, is required for these reactions to occur. We have to have the proteins together, so if we made mutations that cause them to fall apart from each other, we got no integration. And having a plasmid supercoiling was very beneficial for the reaction, meaning that the plasmid had to be under some kind of tension for this to work. And so looking through we started thinking, well, chemically, how does this work? And James was reading through the literature, and there were two primary ways that enzymes like transposases and HIV integrase and things like that operate. One is through making a covalent intermediate with the protein, typically through a tyrosine residue, like this example shown here on the left. And the other way they work is by activating the 3' hydroxyls on the ends of the DNA substrate, so they can actually directly chemically attack the phosphodiester bond in the DNA. And James made mutations in some tyrosines in Cas1 that we thought might be close to the active site, and they had no effect on this reaction. So we thought, well, that first model on the left is probably not what's going on. And then he did experiments in which he tested the requirement for 3' hydroxyls on these DNA substrates. And here's the upshot of those experiments. So this was initially using single-stranded DNAs. We saw no reactions. Here's what I showed you before with a double-stranded DNA radio-labeled, so we can see the products that are forming. And then if we blocked the ends of the DNA with phosphate groups, no products. So that was really an important clue that you need to have those 3' hydroxyls for the reaction. And if you blocked one side of the DNA but not the other, you got a reduced amount of product. So it seemed very consistent with the idea that the way this integrase works is by activating these 3' hydroxyls to form some kind of intermediate that might look something like this. And that's what we're detecting up here on this gel system. And James went on to, he was very frustrated because we've been trying to crystallize the integrase with a DNA substrate, and we had no success. And James has a very good hand, so I knew it wasn't his hand. I knew there was something else going on. And so he did an experiment, and sort of in thinking, using that chemical insight, he thought, well, maybe those 3' hydroxyls on the DNA ends need to be really accessible by the integrase. And so he started making substrates that looked like this, DNA molecules that would have 3' overhangs on the ends. And when he tested those for in this in vitro integration reaction, he found that he got a big increase in integration products when he used molecules that had several overhanging nucleotides on the ends. And the best was really having 5 single-stranded nucleotides on each end of this DNA substrate. And so that allowed him to then trap a complex very stably in crystallization, and you got a beautiful crystal structure of the integrase bound to double-stranded DNA, as you can see right here. And this was a really great moment because we could see exactly, we learned a couple of important things from the structure. First of all, it explained why any DNA sequence can be integrated into the genome, because there were no sequence-specific contacts between the protein and the DNA. And it also showed us why you benefit from having these single-stranded ends on the DNA, because these actually have to code into the active site of each Cas1 protein on either side of the complex. And if they're not single-stranded, double-stranded helix there, a duplex there, it's very hard to get them in. I just want to show you a couple of other things. I'm almost done. First of all, we wanted to know where are these integration events actually occurring in that plasmid that I showed so we used DNA sequencing to get at this, and we found that these integration events occur all throughout the CRISPR locus. And that was, on the one hand, really cool to see that they're really localized to the CRISPR locus, but it's different from what happens in bacteria. And this is just showing you where these actually happen. So they're exactly occurring in exactly the junctions between the repeats and the spacers, but in bacteria, all of the events, almost all the events occur only right here. So there's kind of one growing end of this locus next to this blue sequence called the leader sequence, so that events occur very precisely on one end. So the question was, what is directing that integrase to that site? So we thought something might be missing in our in vitro reactions. And so James took a look at that leader sequence, and he realized that it's a really AT-rich sequence, and there's an interesting protein in E. coli bacteria that binds to AT-rich sequences, and it also bends DNA. And it also plays an important role in various kinds of transposases, things that alter the DNA structure. This is a protein called IHF, or integration post-step. And so by doing a footprinting experiment where you bind the protein to the DNA and then you expose the DNA to cleavage agents, you could show that you get very nice protection of the DNA when you have IHF present in the reaction, and not when you use a mutated CRISPR sequence that doesn't bind to IHF. So that shows that IHF is likely binding right here in this leader part of the CRISPR locus. And then we showed that if you make mutated bacteria that don't have this IHF protein, then you actually don't, you lose the ability in cells to actually get integration into the CRISPR locus. So this is just an autologous gel system that shows when we have wild-type bacteria, we can see over time that the CRISPR locus expands. And we're using a PCR-based reaction where we just amplify that CRISPR sequence, and we can see that it gets bigger over time as spacer sequences are inserted. But if we have these mutated versions, either a dead version of Cas1, the integrase, or bacterial cells that are missing the IHF protein, which has two different subunits, is what you're seeing there, we lose that integration ability. This was done by James together with an undergraduate, Lawrence Bai, in our lab, and another student, Lucas Harrington. And furthermore, if we add back IHF into the cells, now we've restored the ability to acquire sequences. So it really looks like that protein is really important for integration. And furthermore, if we now go back and do the sequencing assay, we now see that when we have IHF present in our in vitro reactions, in our purified system, we now see that almost all of the integration events occur where they should, next door to the leader sequence. And there were just a couple of follow-up experiments that showed that we can get integration into linear plasmids. We no longer need supercoiling if we have this bending protein around to bend the DNA. Furthermore, we can activate a dead CRISPR locus that had mutations that didn't bind to IHF by making mutations that would allow this IHF protein to bind. And now we see restoration of the ability to acquire DNA sequences. And that's what's shown over here on the far right. This acquisition of DNA into sequences that have this mutated leader that can bind to the IHF protein. Sequencing to confirm that. So here's our model. We think that this IHF protein binds and bends the DNA and somehow allows that integrase to start inserting DNA at the place that this single site that's right next to the leader sequence and that first repeat. And that allows the DNA to be integrated. And check this out. It's really a cool mechanism because the DNA goes in, and now you have a single strand of DNA on either side of this inserted sequence that allows it to be copied, and that's how the repeat is introduced, and you recreate the architecture of that repetitive locus by having this little template on either side that generates a new copy of the repeat, flanking this integrated sequence. So, to summarize, we know that CRISPR systems can be harnessed for RNA-guided genome engineering, as I showed you. We know that the Cas9 protein has naturally evolved for very rapid and accurate DNA target recognition, which makes it a useful technology. And finally, we understand now how this integration of new sequences works in the genome, and we think that that also could be an interesting technology for doing things like tagging genomes, something that we're currently working on. I just want to close with 2 slides. I want to share with you that we have a very exciting initiative here at Berkeley together with our sister across the bay, UCSF, called the Innovative Genomics Initiative. We're located right now in Yikashing, but we're moving to the EGI building. And check us out on the web. We're doing lots of exciting science, and we're welcome you to come and visit. And finally, I want to thank a great group in my lab. So I took my whole lab to Hawaii 2 years ago. And I can't guarantee I'll do that every year, that's for sure. But I took everybody back to my hometown where I grew up and first got excited about science. And I took everybody. I took all the undergraduates and all our summer students and our technicians. Everybody came. It was really fun. And so huge thanks to the folks in the group. And then I mentioned several of our collaborators along the way. Science for us has always been collaborative, so I love that about science. Working with different kinds of labs, really fun. And we're funded by a number of sources, but we had really key support from the National Science Foundation that gave me a small but critical grant to get started working on CRISPR systems before anybody knew they would be interested in this. So thanks a lot, and I'm happy to answer questions.
So I know there's a lot of questions. I'm going to ask one of the co-chairs of the Active Education Committee to come and get the mic. And if you have questions, Professor Doudna can select you and ask your questions. Please make them short and brief. Thank you.
Your question, please just raise your hand.
I was wondering, why do bacteria integrate honey bacteria back into their genome? Why is the integration process?
So, can I shout? You guys hear me? Yeah. So the integration reaction is how they adapt to new viruses. So when they encounter a new virus, they steal the sequence from it and insert it into that CRISPR locus, where it can then be copied into RNA and used to guide Cas9 or other related proteins to find and cut that DNA. They find that. That's how we adapt to particles.
Hi. I was wondering, how does it determine which part of the viral DNA is cut up and left in?
Yeah, how does it determine which part of the viral DNA to recognize, right? And we don't really know for sure how that works. It looks like it's mostly random, actually. And the only criterion is finding sequences that are next to that little PAM, that motif that I showed you, the GG. So it looks for that. And what I didn't tell you is that that's part of the self versus non-self mechanism, because when those pieces get integrated, they go in with no motif in the repeats. So they're not identified in the CRISPR locus, because otherwise the bug would just cut its own chromosome, which wouldn't be very useful. So they don't get recognized there, but only in the natural viral sequence where the GG is located. Does that make sense?
Hi. Some of the proposed applications for CRISPR would be like to address like genetic diseases in living people already. Or would it require delivering the Cas9 protein into the cell in some way folded form? Would you be able to potentially like instead use like a virus or something to deliver the DNA forward undecoded in there? What else do you know about that?
Yeah, so you identified a really key step in this technology for applications in human cells or frankly in plants or anything else, right? It's the delivery. How do you deliver these molecules into the cells where you want editing to happen? And for human therapeutics, it's a really big challenge. And it's a challenge not only for this technology, but frankly for any therapeutics. How do you get it to the cells where you need it to act? And right now, the way this is working for Cas9 is there are a few different ways to deliver it. One is, as you just said, which is to have a pre-assembled protein RNA complex that you can then introduce into cells. And that's actually what our lab and several of our collaborators are doing. We think very good at that. But there are other ways to do it too. You can also use a virus to introduce it. So you have it encoded in a viral DNA molecule to deliver it, depending on what you want to do. Yeah.
What's the general overview of the evolutionary process that has kind of evolved from leveraging accurate DNA?
Okay, so she's asking, I think you're kind of asking why. Why did it evolve for rapid DNA particle? I think it's because, you know, if you have bacteria and you're infected by a virus, you've got not very long before you're going to be lice, and that means you're dead, right? So death is a very strong selection. So it really just has forced that system to evolve to be really, really good at finding matching sequences and cutting, because that's, for many bacteria, that's their primary defense against viruses. So, yeah.
So a large part of the intersection between computer science and CRISPR research is applying machine learning to determine which IRA to use. Is there any other application of machine learning that you know of to CRISPR?
Well, okay. So I was at Stanford yesterday, and I was talking to a student who was bound for medical school, so he's a pre-med, and he was saying that he was thinking about this, and he was thinking that, you know, he asked me, would it be possible at some point to have a computer algorithm that will be able to tell you, if I want to make this particular change in this DNA sequence of a cell, this kind of cell, how do I do it? Which guide RNA do I use? Which Cas9 protein do I use, because there are many? How do I deliver it? And how do I ensure that I'm getting exactly the kind of editing that I need for my application? Like, let's say I want to fix the cystic fibrosis mutation. It's a point mutation in the gene. I need exactly the, you know, like one letter to change to another. I need to ensure that almost every cell gets that change. And I think that's coming. It is. We're not there yet, but I've been to Google recently, and I was talking to Google about the possibility of using what they're good at doing, search, right, to aggregate lots of data sets from scientists globally who are doing genome editing on different kinds of cells